So the exercise is around a tiered testing strategy. Can you see it yet, Hedvig?
Yes.
Yes, okay. So as been mentioned, just the sheer number and variation in nanomaterials that are currently on the market or under development presents a real challenge for hazard assessment, because it would just be not ethically or it just wouldn't be feasible to fully assess all of the nanomaterials using long-term in vivo studies. So one approach that may be used to address this would be to apply a tiered testing strategy, which would allow us to filter nanomaterials that have a low hazard potential for ones that are more likely to pose a hazard. And higher tier testing can also provide a higher degree of scientific confidence in your hazard assessment. We also recognize that different stakeholders may have different purposes for why they want to conduct a hazard assessment. And a tier testing strategy also allows you to balance the potential cost and of conducting your testing against the information requirements that you need to most efficiently make a judgment or conclusion on the potential hazard posed by the nanomaterial of interest. So the first question we'd like to ask you is if you could suggest different stakeholders or different users who may require hazard assessment of nanomaterials, and then to consider if all of these purposes that people might be doing hazard testing for need the same degree of scientific confidence, and if not, what tier level could possibly give them the answers that they around the hazards that would allow them to make a decision on their material of interest. So within our tiered testing strategies, we've sort of simplistically divided it from tier 1 containing sort of in silico or in chemical methods and modeling methods that might allow you to predict a hazard. And then you could also employ some simple in vitro screening assays at this level. Where you progress to tier 2, you could employ more complex in vitro models. And then finally, you would have filtered out a select number of nanomaterials for tier 3 testing, where you would incorporate your in vivo models and full regulatory testing strategies. So I can't see the discussion group or discussion forum, but if the other presenters would like to comment on any suggestions that are inputted by the participants, and then we can discuss some suggested answers in a few minutes.
I do not see any suggestions yet. So if there are, I see something here.
Yeah.
That it depends on the type of assay included in the testing strategy.
Mm-hmm. But for the first question regarding different users, so we have some people taking part that are from different backgrounds, so from academia, and I think some people said they're from industry. So the purposes that you're conducting your hazard assessment might be different for these different stakeholders. So do you think everyone needs to know for their nanomaterial of interest what the in vivo hazard posed or the results of a 2-year in vivo, long-term in vivo carcinogenicity study is, or if they would be interested in looking at toxicity or a potential hazard at earlier tiers, and that would be sufficient.
Hi. Sorry, I think I missed a little bit part of the question that Fiona was trying to address here. So you're trying to ask if all these tiers require similar level of scientific confidence in the data that is generated or the assays that are used.
It's whether each tier will progressively increase your confidence in the hazard assessment. But
I think I would say yes, because the lower tier are more like yes or no kind of a question, in the sense, do they induce any response in the trajectory of a disease path? And then more complex models or in vivo models then will allow you to see the manifestation of that particular path, the disease path, into a disease or an adverse outcome. So yes, I guess in my opinion, yes.
I'll move on to the next. So the next slide is just to give an example of some of different purposes someone might be different stakeholders might be conducting hazard assessments, and how these can align differently with the tiers. So as Hedvig briefly mentioned, if you were interested in incorporating safety by design, so if you're a product developer and you have a number of potential nanoforms that you're looking to further develop, you might want to know quite quickly and make a rapid decision about whether your nanomaterials may pose a hazard, rather than actually confirming a hazard through long-term in vivo studies. So at this stage, using these screening assays or predictive models and modeling approaches might be sufficient for you to make your decision. So you actually don't ever need to progress through the tiers. You're happy enough with just doing more simplistic hazard assessment. And similarly, if you were concerned about maybe exposure to nanomaterials within your facility, you might take a worst case scenario or a precautionary approach and just compare, using simple assays, your nanoform or nanomaterial with ones of known hazard. And if they're shown in simple in vitro assays to also cause cytotoxicity or pro-inflammatory responses in acute assays, you might just take then the precautionary measures that are employed for this other nanoform that has undergone much more extensive testing, and just directly apply that to your one without actually conducting the testing. It might be more cost effective for you just to do that, rather than to invest in understanding the full hazard presented by your particular nanoform. However, as we said, as you progress through the tiers, you do increase your understanding of the hazard posed. So if you're interested in getting a better mechanistic understanding of your materials, the tier sufficient, because they would be largely based on much broader measures of a hazard response, rather than the pathways that might be involved or the interactions between different cell types in your complex models. And then finally, if your purpose for testing was for regulatory reasons or to inform a risk assessment, you're most likely to still require some in vivo testing at this stage, whereas the information generated at the earlier tier levels would probably not be sufficient for you to make a confident judgment in the hazard posed by the nanoform. So then also briefly, would like you to sort of consider, if you were to generate a tier testing strategy for nanoforms, what different tests or example models or assays could you include at each tier? And would you set particular criteria to differentiate between your tiers? Or how would you go about generating a tiered testing strategy? And we'll just include some examples and some prompts for each tier on the screen here. But if you have any particular examples in mind, or models such as the one that Sam presented, which tier do you think that should fall under, or would you put that under?
Yeah, so you can again add something to the discussion forum. I see a comment here from Magali Foret. The comment is about that a high level of confidence from the in vivo assays that remains requested for regulatory approval. So hopefully the development of in vitro validated methods can be developed for endpoints like carcinogenicity and specific tissue damage. I think this, we can all agree on that. That it would be very helpful to have validated in vitro methods or a test battery that we can rely on for more complex endpoint, and that this can also be used for regulatory approval.
Just having a thought. Do you think we could use some of the AOP frameworks to highlight or indicate maybe a couple of kind of in vitro based assays that would be targeted towards key events that could form like a toolbox of predictive, I guess, assays for, say, like a tier 1 level. So I don't know, depending on the application of maybe the nanomaterial and how much of the material is actually released into the environment or will come into contact with humans, then maybe it, say if it was well, I'm going with this, say if it was a material that wasn't highly released or was highly unlikely actually to have direct interaction with humans, if we had enough faith in our toolbox of in vitro tools early on, so say in tier 1, and we then saw no outcome from that in terms of any indication of toxicity, do you think then that would be enough to kind of give that indication of that could be a good, safe by design material to then take forward? Or do you think we'd still have to go through all tier processes regardless of the application of the material at the end of the day?
I think it would depend on, so if you say safe by design, so if you're coming at it from a manufacturer's point of view, it would depend on how desirable the characteristics were. If it was something, you could identify something at an early tier that then you could switch out in terms of your material characteristics that would prevent hazard at that stage, so you don't have to worry about it. That would be a nice way to use this. I don't know about the generating sufficient data of an absence of effect, if that would ever be convincing at a tier 1 level. I think that would be a bit of an issue to try and convince someone that when you take the more complex models and things like fate, biopersistence into account in an in vivo situation, how you could predict that at a tier 1 level. So for example, if you consider the low toxicity or poorly soluble low tox materials that have been shown just because they biopersist in the lung and dose builds up to an overload level, that they might actually cause a hazard response at that stage. Whereas in a tier 1 in vitro assay, you might not see them generate any hazard response or cytotoxicity.
So at this stage, we'd still need in vivo testing quite heavily, really, I suppose. At the end of the...
I think you would for absence of reactivity or response. I think for where it would be much easier for the other situation that you'd be able to predict or pick up a hazard with in vitro assays, and then even to the point where if you understand the mechanism through in silico testing or modeling, which is something that we kind of can see with things like high aspect ratio nanomaterials and the comparison to pathogenic asbestos fibers and things like that. If you have a very defined hazard that you're trying to predict, it's easier to work back and then use simpler models and identify this hazard potential at earlier stages.
Yeah. There's one comment in the forum from Jose. You can build a good testing strategy using the first 2 tiers as a starting point to determine the possible general toxicity. That could be like including oxidative stress and inflammatory response and nanomaterial interactions. But he thinks it also depends on the use that the nanomaterial will have. I think this is a very valid comment. I think in view of time, maybe you can wrap up the exercise, Fiona. Then I can try to give the audience a couple of final questions that they can answer online.
So these are just a few suggested answers. So at tier 1, you could use structure-activity relationships to model or predict your hazard, or acellular assays that look at very specific physicochemical characterizations, or then simple in vitro studies, high throughput systems, 2D culture, things that would allow you to very rapidly make a hazard assessment using broad spectrum toxicity markers. But then if you want to understand the mechanism a little bit more, you might, for your specific context that you have in mind, so if it's an inhalation exposure, you might consider using the ALI, or if you're more concerned about potential hazards in the liver or gut or something, you might use models closer to what Sam has presented. You might also want to consider moving towards co-culture models or microtissues, and this will allow you to assess endpoints that might relate to specific key events, or also to probe key event relationships using multicellular systems at this level. However, it's important to point out that tier 1 assays might be quite heavily used and well characterized, whereas at the moment, a lot of tier 2 assays or assays that might fit into the tier 2 level are under development and will still require a lot of validation. And then at tier 3, we did suggest at this stage you would consider using in vivo models. And this could also be using very short-term in vivo models to allow you to address, to identify a hazard, or then to full-on complete large-scale test or longer-term testing, such as OECD test guidelines, which might be up to a whole life or 2-year study in rodent models.